Hello everybody. Today there will be a short deliberation on different evaluation methods for pulmonary drug delivery systems which are applied in its quality control. So this test or quality control methods, we have several set of tests, and depending on the type of pulmonary drug delivery, we have to select the appropriate test methods. Let us first see the flammability and combustibility test. So this flammability and combustibility testing, it is particularly applicable for those aerosol products which contains propellants like some liquefied gases, hydrocarbons or chlorofluorohydrocarbons. So for this test, the first method is flash point determination. So for this, an apparatus is used which is called as take open cup apparatus. So here first aerosol product is allowed to chill to minus 25 degree Fahrenheit. Then gradually the temperature of the aerosol contents are increased, and a particular temperature at which ignition takes place, that temperature is considered as the flash point of the aerosol. The second test is flame projection test. So here inside the chamber where there is a small flame, to that small flame means we know the dimension, means length of the flame. Now to that flame the aerosols are spread, and increase in the length of the flame is determined. So this is called as flame projection test. Then routinely we need to determine some physicochemical properties of the aerosol products like vapor pressure. So for the determination of the vapor pressure of a aerosol product like a metered dose inhaler or dry powder inhaler, we may apply different type of pressure gauges. So the pressure gauge simply will give the pressure, internal pressure of the aerosol container. Then density of the product, so this we can determine by using different hydrometer and pycnometers. Then determination of moisture in the sample. So this is particularly important for dry powder inhalers. So in dry powder inhalers we require free flowing moisture free small particulate matters. There is fine powder of below 5 micron size. For this, routinely we apply Carl-Fisher method, which is a very efficient method for determination of traces of moisture in means adsorb moisture on solid particles. So in this method, we can determine the percentage moisture content of the dry powder. Then also we can test the moisture content by using gas chromatographic technique. So in a gas chromatography, we can see the moisture peak, and its peak height will give the, or peak intensity will tell us the percentage moisture content in the sample. Then identification of the propellant. So we have different ranges of propellant. Now out of this, which propellant is particularly present in a aerosol container, or whether there is some mix up with other propellants, that we can determine by using again gas chromatography. So if we have the standard peaks for different propellants, then easily we can identify. Means we have to match that sample peaks with some standard peaks. Then also we can apply IR spectroscopy for determination or identification of the propellants. Then another important area is evaluation of the performance of the aerosol product. So first is the aerosol valve discharge rate. So at what rate the dispensing is taking place, that we need to know. So for this we can dispense certain numbers of doses from the container, but before this we have to take the weight of the aerosol container, then dispense some fixed number of doses. Then again we take the weight of the aerosol container. Now the weight difference we will get, so that weight difference we can correlate with the time and per unit time also, so that gives the discharge rate of the aerosol valve. Then the spray pattern. So spray pattern means so in what way the spray is going out of the valve. So we need a very uniform, well dispersed droplet or particulate matter from the aerosol valve. So this can be tested by spraying the aerosol on a piece of paper which is previously treated with some dye and some inactive material like talc. So when the spray is applied on the paper piece to the regions where that sprays fall or the spray strikes, so in those areas the dye will be dissolved and it will produce a color. Okay, so that gives a pattern, means how that spray is uniform or not, or all the spray is going to one direction or it is very well dispersed, so that we can visually get some information from this paper. Then doses with metered valve. So for this we can simply take the weight of the aerosol container then dispense some several numbers of doses, say 10 number of spray. Now this 10 number of spray, if we assay it, then the drug content we can determine. So similarly, for others, for 20 number of doses, what is the drug content, that also we can determine. So like that, the average number of dose we can determine. That is the difference in the weight divided by the number of doses dispensed gives the average dose. Then net content is a simple test. So here we have to take that weight of the empty container, then the weight of a filled container, then we'll get the content inside the weight of the contents inside the aerosol container. Then there are some tests on particle size determination. So this is very important for aerosol products. So particle size determination here, one method called aerodynamic particle size. So this aerodynamic particle size is important for aerosols. So here one important equipment very commonly used and prescribed by different pharmacopoeia is cascade impactor. So these cascade impactors are having one input line and output line. So we can imagine how we carry a particle size analysis in a shift shaker. So it is also little similar to that, but here some air nozzles are fitted. So these air nozzles are arranged in such a way that the bigger nozzles, air passes through the bigger nozzles first, then it falls on a platform or a stage, then again a next set of nozzles which has a lower diameter or small size, again it will fall on a stage, so on. Okay, so means gradually the diameter of the nozzles will decrease towards the bottom, and as the nozzle size decreases from top to bottom, there will be increase in velocity of the air. Now through the input, the entry air will carry the sample of powder, means our dry powder inhaler, and it will fall on the first stage through the bigger nozzles. Now the smaller particle will move to the next nozzles, and again it will fall on a stage there or a collector plate. Then again the next particle will move to the next stage. So like that the particles moves through different sizes of nozzles. So these cascade impactors are designed in such a way that it can analyze the particles in between zero point 0.4 to 10 micrometers. It means the biggest particles which are on the top this one platform, that is above 10 micron, and the particles which are written on the stage at the very bottom, it has the capacity to hold particles 0.5 micron, 0.4 micron and above. So any particle which are smaller than 0.4 micron will move out of the last platform, and this is taken up by the that output air, which is the output is fitted with a vacuum. So vacuum will take up all the airs and that filtering small particles. So from this we can differentiate the particles at different platforms or stages. Now we can extract, or we can recover the particles which are categorized depending on their particle size, and we can perform an assay by dissolving the particles in a suitable solvent, and we can find out the drug concentration by suitable analytical methods like UV or HPLC. Then some in vitro test methods are routinely applied. So these in vitro test methods basically uses different cell lines, but cell lines are very limited for pulmonary drug delivery testing. The most popular one is the pneumocyte type 2 pneumocyte 549. So this cell line it is obtained from the lung tissue of human lung tissue, and this is used for different metabolic and toxicological studies, and other, like the absorption, surface absorption, surface dissolution. So this type of studies can be performed using this cell lines. Then continuous cell cultures. So some continuous cell culture, these are the supplementary reproducible and easier to utilize than primary cell cultures, but they lack the differentiated morphology, and also they have a different metabolic characteristic and biochemical characteristic in comparison to our original tissue. And then also these cell lines are very commonly used, and on the surface of the cell there are some cell lining fluids, and particles can be directly applied to the cell surface, where it gives an opportunity for deposition and dissolution in a very small volume of this cell lining fluid. So from that different we can calculation we can make, and this gives very important information about the deposition and dissolution of the particles in different regions of the respiratory tract. Then passive inhalation. So this is also a test carried out for aerosol products. So passive inhalation means that there is inside a hall, a person is taking a aerosol. Now some portion of the aerosol may be distributed in the air, and some other person nearby may also inhale some small amount of the aerosol. So for this determination of passive inhalation, the aerosol product is sprayed into a chamber where mouses are kept, and in the environment of the chamber, the aerosol product is sprayed. Now at different time intervals we can have some atmosphere sample, means air sample from the this one inside the chamber, and we determine the drug concentration in that atmosphere, so that we can correlate with the that how much amount the animal is taking up with time. So if the concentration of the atmosphere decreases due to the inhalation by the animal, that indicates that there is a chances of passive inhalation of the aerosol. Then the certain as that some aerosols are for application on the body, some aerosols are only for application to the respiratory tract. So that is why the application methods we can develop, or there are commercially available chambers where some chambers where we can control the delivery of the nebulizer or metered dose inhaler, and inside that animals can be kept in a controlled condition, and we can analyze the exposure of the drug onto the skin of the animals. So if any that skin deposition and absorption takes place, that we can study. And only head or neck exposure, that also we can carry out. So for that we require some special cases where animals are hold, and only the nose or the head is exposed for aerosol application. So we can apply the this aerosol only to the nostrils or to the near to the mouth, and the quantity which is inhaled, that we can analyze by a suitable analytical method, like say deposition, or other some test we can perform by accurately applying the dosage form to the nose of the animal. Then there are some methods where direct intratracheal administration is carried out. So direct intratracheal administration is more accurate method. So here with some the insufflator, or using some cannula like device, we directly deliver the aerosol product into the trachea, and after different time intervals we can have some sample, or we can analyze the tissue for the deposition of these aerosol particles. So these are some short discussions on the evaluation test for this pulmonary drug delivery system. And there are some newer developments also happening in the field of evaluation of pulmonary drug delivery system. So for today this much only. So in the coming videos I will discuss the newer techniques which are nowadays different research organizations and industries applying for evaluation of the pulmonary drug delivery system. Hope you will be benefited from this video, and I'll be happy to answer any queries